This is the Master Brewers Podcast, brought to you by the Master Brewers Association of the Americas, a volunteer organization dedicated to continually improving the products and processes of our membership since 1887.
Let's go!
Master Brewers brings you interviews with the industry's best and brightest in brewing science, technology, and operations.
Calm down, I'm moving too fast.
Support for this podcast and the following messages come from our friends at the Paul Mueller Company and Hopsteiner. Please thank them. This podcast wouldn't be possible without their generous support.
Paul Mueller Company has been manufacturing quality brewing equipment since 1964. Our innovative design and engineering will save you time, labor, and ingredients, sending money back to your bottom line. Learn more about our new mobile hop module at paulmueller.com. This Master Brewers podcast is proudly sponsored by Hopsteiner, a global leader in the hop industry focused on quality, sustainability, and innovation in new hop varieties and hop products. Contact our brewery sales team to provide you with the hop-related tools you need to craft your next great beer. For more information, visit hopsteiner.com.
So we're able to see individual nucleus inside this long chain of pseudohyphae. And the software from the Cellometer X2 was able to pick each one of those up.
This week on the show, if you use Brettanomyces in your brewery, you know that counting Brett cells is complicated. How would you like to automate that process?
Hi, my name is Leo Chan, and I'm from Nexon Bioscience. I am the R&D manager in the Department of Technology R&D.
Leo, you were here on the show way back on episode 7, where we talked about the poster you presented at the last World Brewing Congress. For those listeners who aren't familiar with your publications, give us a brief overview of your previous work with automated yeast cell counting.
Thanks, John. So the previous poster and publication we have done with MBAA was publishing some work on measuring yeast viability and vitality using an instrument that we have called the Cellometer X2 image cytometer. So what this instrument does is it takes multiple pictures of the yeast that's in your sample, and basically you can take a sample and put it in this microfluidic chip. And using the Cellometer, you can take multiple pictures, and using a fluorescence stain called aquinine orange and propylium iodide, we can stain the live and the dead yeast, and then counting the total number of cells as well as the viability. Then in addition to that, we can also use something called CFDA-AM to measure the vitality of the ale and lager yeast. So by doing this, we can figure out if the yeast are still enzymatically active. So what we have discovered in the past is some yeast, although they are quote-unquote viable, but they actually lost their enzymatic activity, so they're actually not doing anything during fermentation. So by using this other stain, we were able to figure out if they're actively fermenting in a beer product.
And you also did some work where you could really determine some of the other physiological properties. You were looking at, lipids and trehalose and all this other stuff as well, right?
Yeah, exactly. So that was a good point. So we also have done in the past some investigation into how we can measure other parameters from the yeast that people have. So we have tried 3 different things. One is using Nile red to stain the neutral lipids. We also used a label to measure the Trehalose content, as well as the Glycogen content using aquaflavin. So these are basically a direct translation from another paper that we read in the Brewing Institute, Institute for Brewing, and it was done in a microscope setting. So we carried it over to the Cellometer to automate that method. It seems like more people are interested in looking at the glycogen content at this point, from what we have experienced in the field so far.
Very good. And now you've got a new publication on the topic of counting Brettanomyces with a cellometer. What were the challenges of assessing Brett?
So some of the challenges, before I get into talking about the paper, I just wanted to give credits to Brian Martiniak, Jason Bowden from the University of Maine, and which are our collaborators on this project. So they did all the brewing experiment, and then they did the counting, and we worked with them to develop the method to do this paper. So going back to the question about the challenge of measuring Brettanomyces. So I think when we got started on this project with Dr. Bowden, one of the things that we were looking at is how do we measure a yeast that is forming a chain or some sort of pseudohyphae. And I asked them, how do they currently do it? So they didn't have a good answer at that point because they purchased it from Y Lab or Y Yeast, right? Yeast providers. And usually they provide like some sort of density value, but the question is, how do we measure those, right? So it's very difficult if the yeast starts to form the pseudohyphae. How do you count it? And we were trying to figure out a way to really count those, because in the bright field, you can't really see individual nucleus in the pseudohyphae, which is...
Leo, why don't you take a minute to explain pseudohyphae in a little bit more detail? Is that the same thing as pseudomycelium?
It's very similar. So the pseudohyphae is just an elongation of the yeast itself, and when it buds, it doesn't separate; it continues to attach to each other. So you have this long chain of yeast, but they're all connected. So it's very hard to separate out which individual nucleus from within that pseudohyphae. Does that make sense?
It does. And sometimes you can see multiple nuclei in what you think is the same cell there, right?
Exactly. So at least under the bright field imaging in a microscope, you can't really tell and separate out. You know that there's a chain of yeast there, but you can't really tell where each individual yeast start and end.
So that's a pretty big challenge. So how did you go about approaching that challenge of dealing with cells that are so difficult to separate out visually? So
we started out by saying, okay, if we want to actually count the Brettanomyces, we want to be able to measure all the nuclei in the pseudohyphae. So we started out by using the same stain that we have been using, the acridine orange and propylene iodide. And it seems to stain these Brettanomyces very well. So we're able to see individual nucleus inside this long chain of pseudohyphae. And the software from the Cellometer X2 was able to pick each one of those up. So now the question comes back to us is, do we need to count them as individual cell or as a one long chain of multicellular organism? So which one better represents the sample, right? So we did it both ways. And what we found is that yeast, the Brettanomyces that forms the most pseudohyphae has more discrepancy between counting them as one multicellular organism or counting them as multiple individual cells versus Brettanomyces that did not form a lot of pseudohyphae.
And you looked at, might be a good time to point out, so you looked at several different species. You want to talk about which ones?
Yep, so we looked at 3 different species during the experiment. So we looked at Clausinii, Broxylinus, and Lambicus. So out of these 3 Brettanomyces, Lambicus didn't really form that pseudohyphae. It was more like individual cell, very similar to Saccharomyces. And the Clausinii formed the most pseudohyphae, and where Broxylinus formed like the medium amount of pseudohyphae.
Coming up,
we are looking into how we can measure a co-culture fermentation or co-culture experiment in a batch using the Cellometer.
I'm John Bryce, and you're listening to the Master Brewers Podcast from the Master Brewers Association of the Americas. Here's what's coming up on the Master Brewers calendar. District New York meets at Anheuser-Busch in Newark June 14th. District Philly's annual golf outing is June 15th. The District Michigan Summer Social is July 7th at Fitzgerald Park and Grand Ledge. District Northern California meets at Admiral Maltings in Alameda July 19th. Don't miss the Brewery Safety Webinar July 25th. The annual District Texas Summer Meeting is the weekend of August 3rd in Kerrville. The ASBC MBAA Brewing Summit takes place in San Diego this August. This week is your last chance for discounted early bird registration. Register now at mbaa.com, where you can also view the full calendar of events with more details or find a district meeting near you. Now back to the show. All right. Why don't you set us up for the different experiments that were done to get to these methods?
Okay, so within this paper, what we initially did was we cultured these 3 strains of Brettanomyces in 3 different flasks, and we have a stir on, and then just measure their concentration and viability over that time. And we discovered that each of those strain plateau at a different concentration. So it was very interesting to see how that became during the growth phase, and you could see Lambicus didn't grow as much as the Clausenii, and it could be due to the amount of Pseudohyphae. But you could also see as they grew, their viability remained pretty high. I would say above 80% for all 3 strains. And where Clausenii was the most stable one. So that was the first experiment. We're basically using that growing in a control condition to set up our counting parameters on the Cellometer. So after that, what we wanted to do is we're also able to use a plugin software called Fcs Express. So what we did is we plotted in a scatter plot the minor and the major axis, which is basically the length and the width of the Brettanomyces yeast that's counted in the image on a scatter plot. So by plotting that, whichever yeast have very long major axis, which is the length, versus the minor axis, which is the width, that indicates it's either elongated or it's budding. So we're able to measure using this method the percentage of elongation or pseudohyphae formation in the culture. So after that, what we did is we wanted to see how these yeasts behave in a real fermentation. So Dr. Bolton and Brian was able to take 2 of the strains. So we did the Clausenii and Lambicus, and then run them in a typical 100% Brettanomyces fermentation. So we're able to see, also measure the concentration over the about 40 days for both strains. And we're able to see that there's a difference between how the yeast propagated or grew during the fermentation. We're also able to measure the yeast viability, and we could see that during the entire run of the 40 days, the Clausenii again was very consistent, very high viability. However, after about 15 to 20 days, Lambicus viability started to drop down to about 70% and remain about 70% for the rest of the duration. Then we're also able to measure the budding and pseudohyphae percentage during the fermentation, where, as expected, the Clausenii showed a very high Pseudohyphae percentage throughout the entire fermentation, while Lambicus showed initial budding and then drop to very low percentage throughout the rest of the fermentation.
Was there anything different between the propagations and fermentations? Were you just doing both, as you mentioned, to sort of set up the counting parameters in the first phase, or were you hoping to make sure that the analysis would work at different conditions and concentrations, or was there some other reason for all that?
So the reason to do an initial propagation experiment was for assay development. So we wanted to use basically a controlled system to see what the Brettanomyces look like, and then by doing an actual brewing experiment or fermentation experiment, that allows us to put these parameters that we have set up as a real test for a brewery that may be doing a Brettanomyces count in their facility, and see how it looks under the Cellometer. So they can set up similar parameters and then be able to count them more accurately during the entire fermentation. I think at this point, most of the breweries are still doing a mixed culture. So in a product, they would have either a combination of Brettanomyces and Saccharomyces. So some of the breweries have asked us if we could kind of separate them out. I think if they're about the same size, then it'll be very hard. But we are looking into how we can measure a co-culture fermentation or a co-culture experiment in a batch using the Cellometer.
You read my mind. That was my next question. So you observed some different growth curves during fermentation. Did you want to talk more about that?
Yeah, so we actually looked at, during the fermentation, well, actually, in both fermentation and propagation, we noticed that they grew differently. So we actually went back to to some previous publications, and then they did indicate that our results match a similar trend to what the previous publication have published. So it kind of put us in the right spot. So we thought it was probably real data. It wasn't something like a one-time experiment. So we actually did this experiment multiple times, and we got similar results. So I believe it could be just how the each strain forms that pseudohyphae. And I think when it gets into that pseudohyphae mode, it grows much faster. So if you look at the growth curve in the technical quarterly, you can see that, of course, Calcenii had the most pseudohyphae, and it grew the most. Then the next one was Brasilinus, and then finally Lambicus. And you can see that was the trend, and also the trend of the decreasing in pseudohyphae formation. What I think is one of the questions that we really need all the breweries that's doing Brettanomyces to think about is, how should we count these Brettanomyces, and is there a difference? Because if you do count them as a multicellular organism versus an individual cell, there's going to be some difference if you're using Calcenii, and I would say at least 20 to 30% in concentration difference. So how would that affect your product? I think those are the questions because we don't have a fermentation system here, so we definitely need some insight from the field.
So other than investigating the possibility of mixed fermentations, what else is next? What else are you guys working on?
So that's a very good question. Thanks for bringing that up. So in this year's Brewing Summit, we're actually going to present a poster on counting bacteria, total bacteria for fermentation. So if you're doing sour beer, that's a very good application. So we have set up different staining protocols, so we can use the Cellometer X2 to count all the bacteria that's in your culture before you pitch. So very similar to how we count Saccharomyces. We're also working with, again, University of Maine on using the Cellometer to, at the same time, measuring viability. So that we can use the Cellometer to track concentration and viability throughout the fermentation.
That was Leo Chan here on the Master Brewers podcast. Type Chan into the industry's best search bar at MBAA.com to find Leo's 3 technical quarterly articles, as well as several of his presentation slide decks. I'm really looking forward to the ASBC MBAA Brewing Summit coming up this August in San Diego. It only happens every 4 years, and it's not like any other conference you've attended. The Brewing Summit is 100% the science and technology of brewing. No pep rallies or business lectures, and you'll be surrounded by some of the smartest men and women in our industry. If you can only attend one conference in 2018, this should be it. Register now at mbaa.com.
Just like that one day when we came around then.